Hello everyone, and welcome back to another lecture here on cardiopulmonary physiology. We're going to talk today about exercise and the blood flow response during exercise and the importance of the transportation of oxygen and the usage of that oxygen. So here we have some learning objectives. I may have to split this video up into two separate videos to cover everything. But we're going to understand the hemodynamic response to acute and dynamic exercise and understand the basic physiological mechanisms that underlie that hemodynamic response during exercise. We're going to understand how heart rate, stroke volume, cardiac output, oxygen extraction play a role in the response to exercise, and how does our muscle pump help aid to the cardiac output, to the performance of aerobic dynamic exercise. And so we have to transport oxygen throughout our bodies. We take in oxygen from the air. That air goes into our lungs, and that oxygen diffuses across the lung wall into pulmonary capillaries where there are red blood cells. See, the blood itself, the plasma, is not really good at holding on to oxygen. It doesn't have a high solubility. So these red blood cells, or what we call erythrocytes, are the ones that are going to transport oxygen throughout our bodies. And the reason they're able to transport oxygen is because they contain a protein called hemoglobin. And so hemoglobin, the reason for the heme is because it has 4 heme or iron groups, right, that connect to one molecule of oxygen. And so since the hemoglobin contains 4 of these heme groups, then each erythrocyte, each hemoglobin is going to be able to carry 4 molecules of oxygen. And here's the structure of hemoglobin. As you can see, there are 2 alpha chains and 2 beta chains. Again, each of these containing an iron group here in the middle. And so once the oxygen molecule binds to the hemoglobin, it's going to change the shape and change the affinity that hemoglobin has towards oxygen. And this is, pretty high level of detail. It's not necessary to know this, but that iron is going to be surrounded by amino acids called histidines. And so once the oxygen binds to that iron, the histidines are going to sort of lock into place and change the shape of the hemoglobin. And since this is a protein, it's a long chain of amino acids, once that change occurs in one of these subgroups, then it's going to change the structure of the whole entire protein. And by doing that, we increase the affinity for more oxygen molecules to bind. So as one oxygen molecule binds to one of these chains in hemoglobin, the affinity or the attractiveness to oxygen is going to increase, and it's going to be more likely that this red blood cell gets more oxygen bound to it. Again, our oxygen binding capacity in the plasma itself is very low, about less than a milliliter per 100 milliliters of blood. But since we have these red blood cells, that hemoglobin is going to be able to hold on to around 20 milliliters of oxygen per 100 milliliters of blood. So this is what allows us to have high levels of aerobic metabolism, and it's going to be able to make sure that during exercise that we have a large amount of oxygen that's able to be transported throughout the bloodstream and then eventually dumped off to cells, metabolically active cells. So here is it's a elementary way of looking at our circulation, right? The very basics of it. We have the heart, which is responsible for generating the pressure necessary for blood flow. We define its work as cardiac output, and that cardiac output is responsible for blood flow throughout our body. And so here we're going to see that we have mixed venous blood that enters the right atrium, the right side of the heart here. And so that mixed venous blood is the blood in the pulmonary artery. And so that blood that is leaving the right ventricle and heading to the pulmonary circulation in order to get oxygenated, right? And so this blood is going to be sort of blue or purple in color because it has a lower amount of oxygen. It is deoxygenated Doesn't mean it has zero oxygen, but it has a relatively lower amount of oxygen. And then as we work our way from that pulmonary artery to the circulation of the lungs, that blood is going to go in pulmonary capillaries, right? And that blood is going to border the alveolar walls, which are taking in large amounts of air and large amounts of oxygen. And so that oxygen will diffuse through the pulmonary wall to the pulmonary capillary and then start to bind to hemoglobin. And then there's going to be a large amount of oxygen that binds to hemoglobin, and now that's going to become oxygenated blood or arterial blood. And so once that blood is then transported back to the left side of the heart through the left atrium and then the left ventricle, that is oxygenated blood or arterial blood, the blood in all of the arteries. And since this is sort of getting mixed together here on its way back from the lungs to the pulmonary vein, you're going to have that equal distribution of oxygen when it makes its way back into the left side of the heart. And then the left ventricle, is responsible for generating that pressure to eject blood out into the aorta and then into the arteries and arterioles of the circulation in order to supply that oxygen, those nutrients necessary for cells. And so from the arterial side of the circulation, we're going to go into the systemic capillaries, right, where the other cells of our body are. And again, there's going to be that exchange of that oxygen, those nutrients, and then we're going to pick up CO2, the waste products, and then that venous blood gets carried out, and this cycle happens over and over again. And so the way that oxygen binds to hemoglobin, it happens with something called cooperativity. And so as one red blood cell gets filled up with oxygen, there's basically, it's more likely that oxygen is then going to bind to the other red blood cells. And this is especially going to happen when you have higher partial pressures of oxygen around. And so the oxygen content is the amount of oxygen in a given amount of blood, right? And so we usually, the units for that is milliliters of VO2 per 100 milliliters of blood. And so it's going to depend on the concentration of hemoglobin, how much hemoglobin you have in the bloodstream, and then the oxygen binding capacity, right? The ability to bind to that oxygen, the alveolar VO2, so how much pressure of oxygen are you getting into the lungs, the pulmonary diffusion capacity, so how easy is it for that oxygen to travel across the alveolar wall? Do you have thickened alveolar walls in case of someone who smokes or someone maybe with COPD or emphysema? And then alveolar ventilation. How much of that air is actually getting to the alveoli, to those, to that diffusion site, so that oxygen could go through the alveolar wall into the bloodstream, right? And so we call the amount of hemoglobin or the percentage of hemoglobin bound to oxygen, that's called saturation, right? And so if we're in a place where the oxygen partial pressure is high, think about the lungs. This is where the VO2 is probably going to be the highest throughout the whole body. Well, that's around 105 in the lungs, and so that's going to mean that a lot of oxygen is going to bind to hemoglobin when that blood passes by the pulmonary capillaries, right? And so what you see is that, once that deoxygenated blood makes its way to the lungs, most of those hemoglobin are going to be attached to oxygen and then make its way back down to the left side of the heart where it gets pumped out to the rest of the body, right? But then if you look at somewhere in the body with low levels of oxygen, like the tissues, right, the systemic tissues, think about the skeletal muscles that are using up oxygen and utilizing it, there's not a lot of oxygen in that area. So what you're going to happen is there's going to be a dump off, and that oxygen is going to go from a high concentration to a low concentration, right? Now, in the body, not all of the oxygen is going to get dumped off, but a good bit is going to be supplied to the tissues so it could utilize it for metabolism. And so what we're looking at here, the relationship between the partial pressure of oxygen and the saturation of hemoglobin, this curve is called the oxygen dissociation curve, right? because it's going to show you when in time, when is the environment that hemoglobin is going to dissociate with oxygen and that oxygen gets dumped off. Now, this isn't said, but it also shows you the association of hemoglobin to oxygen too, where if it's in a high an area with high partial pressures of oxygen, a lot is going to bind to those hemoglobin. So in the lungs, a lot of that's going to get picked up, and a lot of that oxygen is going to bind to hemoglobin. And then when we go out into the cells of the body, a lot of that is going to be dumped off for those mitochondria within those cells to utilize. And so again, here we look at the circulation and we look at the partial pressure of O2. It is going to be highest in the atmospheric air. By the time it makes its way down to our alveoli, it loses some of that PO2, partially because of humidity. I'll talk about that in future lectures. A small percentage is dropped off due to humidity, and then because we have a larger concentration of CO2 in our lungs than the atmospheric air, that PO2 of the oxygen starts to decrease as we make our way down through the pulmonary tract, right? But still, within our bodies, this PO2 is going to be highest in our alveoli. And so when that blood is transported from the right side of the heart up into the pulmonary circuit, it's going to have a lot of O2 diffuse through that pulmonary wall. It's going to bind to those hemoglobin, and then that arterial blood is going to make its way back. And so when that arterial blood, though, gets pumped out to metabolically active tissues, there's going to be a low pressure of O2 there, and then that O2 gets dumped off to the mitochondria for them to utilize. Whether it be gases, whether it be liquid, they always flow from areas of high pressure to areas of low pressure. So since the PO2 is higher in the alveoli than the deoxygenated blood, that's where O2 is going to travel. And then vice versa for carbon dioxide. That carbon dioxide is higher in the deoxygenated blood, the venous blood, than the pressure of CO2 in the alveoli. So that dictates the diffusion of those gases, right? It's always going from a higher concentration or pressure to a lower concentration or pressure. And so this oxygen, we can calculate how much we actually utilize, right? The cardiopulmonary functional capacity. And so we use a term called VO2. So that big V is going to stand for volume of O2. And that is the volume of O2 that we are actually utilizing and consuming, right? So we're going to consume it and use that VO2. What are the cells actually utilizing as far as the volume of oxygen is concerned? And so in order to calculate VO2, we use something called the Fick equation. And that is cardiac output times what is called AVO2 difference, that delta sign that stands for the difference. So that little a, that stands for the oxygen content of the arterial blood. And that little v, that stands for the oxygen content of the venous blood. So basically, I like to compare it to stroke volume, because in order to calculate stroke volume, you have a before, an end diastolic volume, which fills up the left ventricle. And then you have an after, the end systolic volume. How much blood was left over after the contraction? And when you take the EDV and subtract the ESV, that's how you find your stroke volume. You had a before, you had an after, and you were able to tell how much blood actually left the left ventricle. So that relationship is similar here. So we're looking at how much oxygen is actually extracted, pulled out of that arterial blood. So you have a starting point of O2, that arterial O2, and then you have an ending point, that venous O2, what was left over after the mitochondria utilized some of that oxygen. And so when you find that AVO2 difference, that's basically saying how much oxygen was pulled out, how much oxygen was extracted and utilized and consumed by the cells of the body, right? So here we have how much blood is being transported, therefore how much oxygen is being transported, and then out of that blood, out of that oxygen, what is actually being utilized. And so again, cardiac output is heart rate times stroke volume. That's how we determine how much blood is being delivered. And again, this delta stands for difference, arterial minus venous O2. That's the difference in oxygen content between the arterial and venous blood. And when you think about it, both of these values are going to increase during exercise. We're going to see an increase in sympathetic nervous activity, which will increase our heart rate, our stroke volume, therefore cardiac output. And those mitochondria are going to be working harder. So, the amount of oxygen that we take in is going to be higher. So that's going to make our arterial oxygen higher, and then the amount we utilize is going to be higher. So that venous oxygen is going to be lower. So we should see an increase in that AVO2 difference, in that value. Because as we recruit more skeletal muscle fibers, more motor units, and those motor units work harder, we utilize more oxygen. And so by definition, the limit of cardiopulmonary function is reached at the highest attainable oxygen uptake, otherwise known as our VO2 max. And our VO2 max is basically what we use for aerobic endurance. That's how we define how aerobically fit someone is, right? And so if you want to see, how adept is someone at endurance aerobic activity, you're typically going to conduct a VO2 max test. And so the classical concept of VO2 max is that exercise with small muscle masses elicit low peak values of oxygen uptake, right? And so as you increase your work, this is the real definition of classical concept of VO2. As you increase your work, right, you increase your intensity of your exercise, it's going to be proportional to your VO2 from the amount of oxygen that you are consuming. So if you are, on the treadmill and you're walking, once you take to a jog, that's a higher intensity. You're producing more workage out of your skeletal muscle. And since your skeletal muscle are working harder and consuming more energy and utilizing more of that ATP, they need more oxygen to do so. And so for a VO2 max test, they will raise the incline. They're going to raise the speed of running, which translates to more work. And in a linear fashion, the harder you work, the more oxygen you utilize until, and this is the ending part of the concept, until your VO2 max is reached. So if there is an increase in work, an increase in intensity, and the body cannot physically consume or utilize more oxygen with that increase in work, you've reached your VO2 maximum, right? And so we'll talk a bit more here on the next slide about defining that. When do you know someone has reached that VO2 maximum? And so part of the reason why we use this for aerobic fitness is because it's highly reproducible. And so if I took someone today and I did a VO2 max test, took them through a treadmill protocol, and then a week from now I did the same exact thing, there should be very little variability in between those two measurements, right? And so here we have around 2 to 4% difference in those measurements. And so it's a lot less variable than other measurements that we use day to day. And so again, how are we going to know that someone has truly reached VO2 maximum? Because there's also something called VO2 peak. Maybe you've seen them during that test, they reached their highest VO2, but they actually were aerobically able to handle more. So we use what's called a primary criteria to see VO2 maximum. And so in this scenario, you're going to have an access to a metabolic cart, the mass to measure, through that metabolic cart, how much oxygen is being consumed, how much CO2, the VCO2 as well. And you're going to be able to tell what's going on as you increase those levels of intensity during the test. And so as you increase the work, right, that VO2 in a linear fashion should go up as well. And so that primary criteria, what is that threshold to know, okay, they are at their VO2 maximum. And so that primary criteria, that's going to be an increase in work during a test, an increase in intensity with less than 150 milliliters of O2 per minute of an increase, right? So here we see the person is working here. Let's say they're around 350 watts of work. They're at 10% incline, they're going at 7 miles an hour, right? And then what you're going to do, you're going to bump them up to 12% incline. And so if you do that, right, and then shortly after that, you're seeing that their VO2, which should go up with increases in work, right? If their VO2 does not go up during that increases in work, or it's less than 150 milliliters of O2 per minute, well, then they've reached their VO2 maximum, right? And they continue to run. That test is probably going to end pretty shortly because the body is not physically able at that point to take in or utilize more oxygen. So they've reached their limit as far as aerobic performance is concerned. And so we call that, again, primary criteria. So you're actually physically measuring the VO2, and then you're determining with an increase in work, you're not seeing at least 150 milliliters bump up when they are going through the next phase. So this is one way of achieving VO2. We also have something called secondary criteria. Are they valid? So let's maybe first list them off and then we can explain why they may not always be valid. The 4 primary criteria for knowing that someone is at VO2 maximum. So the first one, number 1, the first secondary criteria, that makes no sense. The first characteristic for a secondary criteria is going to be a heart rate that is within plus or minus 10 beats per minute of their age predicted max. And so the reason why this is a good secondary criteria for measuring VO2 maximum is because typically when someone, just like VO2, when someone increases their intensity of work, their heart rate goes along with that intensity in a linear fashion. And then once you reach your maximum heart rate, you're typically at your VO2 maximum. So it's a good way to see, okay, they're within 10 of what we know is their age predicted maximum. So they're probably close, if not at their VO2 maximum. Then we have another secondary criteria is an RER of above 1.15. And along with that, I'm going to tie in the third secondary criteria, which is a blood lactate level of a greater than 8 millimoles of lactate. And so the reason why both of those are used to sort of estimate VO2 is because of the nature of what they're measuring, and that is blood lactate. And so RER stands for respiratory exchange ratio. And the physiological range, we typically talk about 0.7, which is the organism is running off of all lipids and fats for metabolism. And then we talk about 1.0. So that's the range typically, 0.7 to 1.0. And that 1.0 means the organism is running strictly off carbohydrates and glucose for their metabolism, right? And so at rest, we fall somewhere right in the middle. More aerobically fit people will fall towards that 0.7 range, while more sedentary, less active people may fall towards that 1.0 range, right? Not exactly 100%, but they tend to be more on that scale towards the carbohydrate metabolism at rest. As intensity of exercise increases, we tend to shift to that 1.0 towards carbohydrate utilization for our metabolism with increases in intensity during physical activity. But what happens is, at a certain point, if glycolysis is going on and we don't have the oxygen necessary to carry the byproducts, that pyruvic acid, into acetyl-CoA through Krebs cycle and then deliver those electrons, those hydrogens to the electron transport chain to furtherly to utilize that energy for aerobic metabolism and get ATP, what we're going to see is that pyruvic acid, instead of going down the aerobic pathway, is going to go down another pathway, right? And that pyruvic acid can be converted to lactic acid or lactate, right? And so that lactate, if you don't have the oxygen, right, to keep up with that metabolism, with that increasing intensity of physical activity, you're going to produce more and more blood lactate, right? And if you're not able to break it down and buffer it out and you're still accumulating lactate, well, it's going to start to build up. And that's why during exercise, you feel that burning sensation at high intensities. And so if we reach above, so we said RER, 0.7 to 1.0. Well, where's 1.5 coming from? So as you increase the levels of lactate that you are producing, right, your blood lactate levels are going up, so that was one of the primary criteria, that's going to shift your RER to over 1.0. So not only are we at 100%, carbohydrate metabolism, but we're going past it. So that accumulation of lactate raises your RER, your respiratory exchange ratio, and it raises your blood lactate levels. So it's a good way to sort of estimate, okay, this person is starting to reach their maximum as far as aerobic metabolism is concerned. They're starting to reach the point where they're not able to get the oxygen to utilize all of the glycolysis that is going on. They're not able to take those byproducts and take them through that aerobic pathway to match the work. And so what you see is an accumulation of lactate as that exercise gets harder and harder. And so the last one is also RPE of above 17. And so that's usually a Borg scale 6 to 20. So if the person, their rate of perceived exertion is around a 17 out of 20, or above, it's going to mean that, yeah, they're starting to reach their maximum limit for what they can handle. Okay, so again, when we talk about validity, of course, when you are directly measuring VO2, right, that's going to be a more valid way to do it. And so, that's what we do in order to really determine VO2 max. We hook them up to a metabolic cart, increase the intensity of work, and see where that maximum limit is. If you're relying on heart rate, well, heart rate can be adjusted based on, the things that you consume, the different medications that you're on, certain drugs like caffeine, stress levels, same thing with RPE. Sometimes a workout feels fine one day and then feels like the hardest thing you've ever done the next. So those aren't specifically valid when it comes to working out. It could oftentimes underestimate these values, and you could see that down below. Even though this matches the secondary criteria of 1.15, we see that we're still a good maybe 15% under our actual VO2 maximum. And so if you want to test, did this person just reach their peak during the exercise and maybe their skeletal muscles couldn't handle the work anymore, maybe they couldn't handle the incline or the speed anymore, or did they really reach their aerobic capabilities? Did they really reach their VO2 max? Well, the best way to do that is that the next time when you perform a test, you perform a supramaximal test. So if I was using that example from earlier, the person was at 10% incline, they were running at 7 miles an hour. Well, maybe bumping it up to 12, maybe you bump it up to 13 and you bump up the speed to 8. And so you're taking a bigger intensity workload, right? So you're shifting this more to the right here. And either if they were at their true VO2 max the other day, the same thing's going to happen and you're going to see less than 150 milliliters of O2 per minute of an increase, or it does go up by 160, 200 milliliters of O2. And that shows you, okay, no, they weren't at their VO2 max last time. They just achieved their VO2 peak. All right, so what is going to determine VO2 max in different populations? Meaning that, if you look at the three aspects of VO2, the heart rate, the stroke volume, the AVO2 difference, what really makes up for that difference across populations? So here we see a figure that basically measures those things in three different populations. We're going to have A really athletic person, like an elite level athlete, and so that's going to be the ATH here. We're going to have a normally active subject, right? Maybe recreationally active. They go on a walk a few times a week, nothing too serious. And then this MS, that's actually going to stand for mitral stenosis, which is a condition where that mitral valve is not the normal size, and so that can cause complications as far as cardiac output is concerned. So now when we look at VO2, we're of course going to see that this mitral stenosis, and you could just, just to make it easy, this is just a disease state, someone who maybe has heart failure or heart complications. That VO2, of course, is going to be lowest in this population, followed by normally active people, and then athletes are going to have way higher VO2 maxes. So if these people are the same age, right, then you can predict that their heart rate maximum should be pretty similar. How do we predict heart rate max? It's 220 minus age. So, 3 populations, all the same age, their heart rate max should be relatively similar. And so here we see, after taking these people through a maximum test, that yes, they were all pretty similar once they reached their heart rate max. 190, 195, 190. So all pretty similar within that same range. And then we look at AVO2 difference, or the oxygen extraction, right? And all 3 populations, even though they have different levels of physical fitness, their AVO2 difference, their oxygen extraction, all around the same volume, around 16 or 17 milliliters of oxygen extracted per 100 milliliters of blood. But then the real thing, right? So if this is going to be the same, and this is going to be the same, well, then the difference has to be in stroke volume. And so that's the answer to this question. What determines VO2 max in different populations? It's going to be that stroke volume. And so here we see, taking them through the test, that, with increases in oxygen uptake, with increases in intensity, that athlete is just able to get out more blood out of their heart, higher contractility of their left ventricle, higher amount of blood being pumped out. Therefore, that VO2 is going to be highest in that population. And so we're going to talk about the benefits here being, an athlete and what that means as far as aerobic performance is concerned. But I do want to underline one thing, and that's how does heart rate increase? So as you start to exercise, from light exercise then to higher intensity exercise, how's it increasing? And so basically there is a cutoff line, and we'll use 100 beats per minute. That's our cutoff, right? And so when you go from resting heart rate to 100 beats per minute, that increase from that level is all due to parasympathetic withdrawal. It's not due to the release of norepinephrine and epinephrine at that point. It is due to less parasympathetic activity, right? Through less activity through that vagus nerve, which at rest keeps our heart rate low. But then, as you increase your activity, it's going to stop releasing as much acetylcholine. It's going to stop binding to those cholinergic receptors, and that's going to increase the heart rate, right? So at 100 beats per minute, all of the parasympathetic withdrawal, or I should say probably most of the parasympathetic withdrawal, has been released, right? So, if you want to just look at the control of heart rate, in the lab here, so what these researchers did is that they blocked the parasympathetic nervous activity with atropine, and then they just saw what happened. So when you blocked the parasympathetic nervous activation, heart rate went to 100 beats per minute, and that was it. So with no additional sympathetic input, no additional sympathetic messaging, heart rate went from rest to 100 beats per minute, right? But then at this 100 beats per minute point, all the increases from there to max heart rate are due to an increase in sympathetic nervous activity, right? So that means the release of norepinephrine binding to those SA nodes and those AV nodes. That's going to increase the heart rate, right? So that's how we get from 100 to maximum, an increase in sympathetic nervous activity, more epinephrine binding to beta receptors in the heart, which help to increase heart rate. So at this 100 beats per minute line, number one, we're roughly at 40 to 50% of our maximal oxygen uptake, which is why we like to use heart rate as an indicator of VO2. And then, so we're at 50% VO2 max here. Most of the parasympathetic withdrawal has taken place, and then additional rises in heart rate will be due to sympathetic nervous activity. And so we'll stop it here and talk about this a little bit more next lecture, but we're going to discuss why do athletes have such a high stroke volume, right? What allows an athlete to produce these higher stroke volumes to have such high VO2s, right? So stay tuned. That'll be part 2 of this lecture.